Hi, I'm Youngjun Park. I'm PhD student at International Max Planck Research School for Genome Science with Professor Anne-Christine Hauschild. And the study I wanted to share here is Heterogeneous Domain Adaptation for Species-Agnostic Transfer Learning. In the biomedical research, we are highly dependent on model organisms. Especially in medical research, we use a lot of mouse and zebrafish model. However, it is not easy to transfer knowledge from mouse and zebrafish model into the human research. So the main problem I want to solve here is how we can integrate biomedical knowledge from different species. And main hurdle on that issue is we are having different number of genes. So the current solution is using orthologous gene information. So many of the study they use gene homology or orthologous gene database to convert one species gene ID to the other species gene ID. However, during that process, there is severe information losses. For example, in this study, they only can use almost 4,000 genes out of 20,000 genes in human and mouse. And on top of that, gene homology is not a solved problem. So there are still remaining issues about how can we handle many-to-many mapping of gene expressions. And furthermore, only well-known organism we can use those information. So we can use pathway, or we can use gene ontology, or we can use other functional similarity. However, only a small subset of species that approach is possible. So our approach is using domain adaptation, which is subcategory of transfer learning. And the typical domain adaptation is homogeneous domain adaptation, and they suppose same feature space but different data distributions. However, in our case, we have to use heterogeneous domain adaptation, which suppose different feature space and different data distributions. So one of the approach is using latent embedding method. And here is our baseline model. So we use cross-domain structure preserving projection for heterogeneous domain adaptation. And this is based on the LPP. And the left one is the main objective of the LPP. And if you look at the CDSPP on the right side, they have 3 LPP terms. Each of that is to minimize distance between source to source domain and source to target domain and target to target domain. So basically CDSPP is kind of supervised PCA method. So we will use known labels from 2 different species data and use that to align 2 different feature space of 2 different species. So here is our overview about the species-agnostic transfer learning. So at first, we have to extract feature on each of the species. And after that, we're going to apply heterogeneous domain adaptation method with known partial set of labels on 2 different species. And after that, we can find common latent space, and we can analyze those 2 different species data onto that common latent space. So we're going to evaluate our method with single-cell sequencing data, and the main task could be the cell type prediction across different species. The first result we wanted to show here is a cell stimulation study, and they have 4 different species, and each of the species has 4 different cell types. So we masked one of the labels on target species and tried to predict based on the source species data. So here's the main result. In this case, we didn't use any gene filtering method. We just applied the PCA and extract features and align that on the heterogeneous domain adaptation method and try to predict it. So on the most of the case, we can predict masked cell types of the target species really well. And this is the example of the common latent space analysis. So the first row shows the pig side latent space, and the second row shows the rabbit side latent space. So we can clearly see different cell types on different species. They are located on the same space on the latent space. So based on that, we can easily predict masked cell types from the source domain knowledge. And this is a gene ontology analysis of top 100 important genes on the first axis of the pigs and rabbit. So we can see they have similar terms regarding leukocyte or related to the immune responses. After that, we compared our SATL approach with other generalized zero-shot learning model. So main task was the out-of-sample prediction. We masked 2 classes on each of the data set and tried to predict in the same way. So we used 3 different single-cell sequencing data set, and each of the data set has human and mouse pair. And here we show the balanced accuracy of unseen classes of SATL and other generalized zero-shot learning model, and also some of the heterogeneous domain adaptation methods. And among all the methods, our SATL approach showed the highest balanced accuracy of unseen classes label predictions. In summary, this SATL work is the first analysis using heterogeneous domain adaptation on out-of-sample label prediction across different species. With SATL approach, we can exclude a gene ID conversion step, which has no optimal solution for many-to-many mapping and having inevitable severe information loss. Its ability is highly dependent on feature extraction method. We found that, and this method can be helpful for analogous gene analysis. And thank you.